Good afternoon, everyone, and welcome to the ASA SLDS webinar. The speaker for our webinar today is Professor Peter Song. Peter does not need much introduction as he is a prolific statistician. You saw Peter's article from time to time everywhere. Just to introduction, Peter is a professor of biostatistics at the University of Michigan School of Public Health. He has published over 230 peer-reviewed papers and graduated 26 PhD students and trained 6 postdoc research fellows. A very energetic guy. Peter's current research interests include data integration, distributed inference, high-dimensional data analysis, longitudinal data analysis, mediation analysis, and spatiotemporal modeling with applications in nephrology, chronic disease epidemiology, environmental health services, and nutritional sciences. He is IMS Fellow, ASA Fellow, and elected member of the International Statistical Institute. Peter now serves as the area editor of the Annals of Applied Statistics in the area of medicine, electronic health record, and smart health, an associate editor for JASA, Journal of Multivariate Analysis, and a statistical reviewer for Kidney International. Without further ado, let's welcome Peter.
Thank you, Zihua, for the nice introduction. Very happy to have this opportunity to share some of my recent research project. So let me share my screen and share. Okay, what's this? So here is my presentation slides, full screen. So today I'd like to talk a little bit about smart health. This is kind of research direction I've been sort of considering in the past 10 years or so. I really feel that this direction has a lot of potential to come up with a new statistical method and some good integration with data science and computer science. And I think that you can see my sort of perspective, what I'm doing and what I like to do next, right? So this is joint work with my former PhD student, Margaret Banker. She now is assistant professor at Northwestern School of Medicine, and current PhD, Le Yaojiang. And then this research is funded by NIH-NSF. So essentially, I'm going to talk about how to use optimization algorithm to solve some of the functional data analysis problems motivated by smart health. So I have recently been sort of very much into the news about this sort of the humanoid robots. I feel that this is a new direction that would create some new data that we probably will be will to see in a few years, right? So here is a list of this 15 top companies that are manufacturing humanoid robots, and then I think these humanoid robots will generate unique data that will really impact many our life, including health management and healthcare. So in the list, you can see Tesla Optimus. This is really a leading sort of the company that manufactures the humanoid robots, and that I don't know in 5 years how this will look like, and is statistical community well prepared to really look at the data generated by those robots. So here is actually the recent announcement by Elon Musk about Tesla ambition on production of humanoid robots. Basically said recently that Tesla is going to build at least 50 Optimus robots in year 2025, and then they are going to increase to 50,000, and then a year later are going to build 10 million robots a year or more, right? So this is really a fast sort of the production of the humanoid robots, and that statistic community will probably see a lot of data coming from this kind of the line of innovation. So talking about the, this what this humanoid robot can affect our life, particularly for the healthcare and management. So just use the chat GPT to create this sort of the image in my mind that what's going to happen, and the one a possible application of humanoid in healthcare domain. And I just create this prompt, like senior man who wears a wearable device like a mini robot, right? So that communicates with a humanoid robot, which stands there and hold this tray of its either tea or a kind of medication that provides at-home services of healthcare and health management. So I think that there's quite a bit opportunity for us to really think about how this wearable device will communicate with this humanoid robot, and how this sort of the communication could take place between these two types of robots, right? So as part of this process of health management, healthcare. So I feel extremely important for us to really thinking about this emerging new data types from wearable devices, right? So that how the data could be analyzed and learned to navigate and supervise humanoid robots to work and to provide really good healthcare and health management. I think this will happen, but just a matter of time that statistics community, we need to act and act fast and analyze various of wearable device data, and to gain more experiences how we're going to really be part of this innovation, right? So of course, smart health, mobile health, digital health, robotic health have been sort of uh, receiving and giving people have received attention to those sort of new topics, but I don't think we have paid much sufficient attention to this fast evolving evolution and then innovation. I think we need more effort in this direction. So that's why today I like to choose this topic to share some of my own experience how to analyze wearable device data. It's a very minimal level of understanding the data, but I think this will serve as kind of the demonstration how we should put more effort and our sort of the research interest into this field. I think that in 5-10 years we will see massive data coming, and because of this sort of the uh, innovation, right? So from now on, I want to concentrate on one topic that sort of related to this smart health, and particularly focusing on the understanding of wearable device data, and how we use the wearable device data to answer some of the uh, interesting problems, right? So this is the paper that has been appeared last year in Annals of Applied Statistics. That is essentially the basis of my talk. So one question we're trying to ask here is that is a physical more active person biologically younger? I mean, this is a motivation for a lot of people who do this regular exercise, because they think that if we stay like physically more active, maybe we can be biologically younger, or will have be a improve our can help improve a health condition and so on and so forth. So then this is a hypothesis, like we like to see whether or not that there's such association between physical activity and biological age. However, measuring relative variables in this kind of study is not easy job, right? It's very challenging because first of all, how do you measure biological age, right? How do you objectively measure physical activity? Measuring these two types of variables, okay, is non-trivial, okay. But recently, because of these technology advances, we have this opportunity to at least to study part of the, uh, part of this problem using some of the new biological platforms and wearable devices. And in particular, that DNA methylation, okay, this platform, right, generates genetic variants, particularly this methylated CpG site that have been used by biologists to predict biological age. And then the wearable devices have been widely used to, capture, real-time physical activities and provide, the monitoring of physical activities on our daily life. So this sort of the appearance of the both sort of the technologies would really give us opportunity to really, measuring the relevant variables and then to answer that questions we like. So when we use epigenetic, we use the methylation to predict biological age, right? Then in the literature, this predicted age is often referred as epigenetic age. And then this epigenetic age is deemed to reflect cellular change and evolution, and that is believed to be more suitable to reflect the aging than the chronological age, right? So we celebrate our birthday every year. So then according to the chronological calendar, but that is a surrogate, the marker of aging. If we want to measure the aging process more accurately, and biologists suggest that in recent literature that using methylation data, okay, to capture the change of our aging process influenced by environmental or other biological exposures, and because they are cells and tissues are impacted by those exposures, right? So that DNA methylation would be a good sort of the markers to reflect those changes influenced by environment, social determinants, and other exposures. Okay, so you can see that uh, there's a little, very small heterogeneity for people who have, young age. But if you're talking about, people at the age 70, then you can see a huge heterogeneity. Like, for example, some people are still very active, even they are even able to run marathons at age 70. And some are start to lose a lot of mobility at age 70. There's a huge heterogeneity, and we are thinking about, I mean, what the variables could be, those biomarkers that can be used to really quantify or reflect this heterogeneity over this aging process. So methylation data, particularly for the epigenetic age, have been used, by many researchers to quantify this kind of biological aging process. So there are many epigenetic age. The age for humans, the age for animals, and there are more than 300 biological age and calculated by different types of DNA methylations generated by different platforms and also different phenotypes, right? So in this prediction analysis, and the earliest one is the Haworth clock, right? This is the first clock that Professor Haworth from UCLA used the methylation data to predict the biological age, and the age clock has been validated by a very wide range of the populations and different the types of methylation data. So, and since then that there are a lot of the and advances or research in this field, and now I think there are more than 300 clocks available in the literature to capture different types of the aging process, like cognitive function, organ aging and tissue aging, all or skin age or so on and so forth. So there are a lot of research going on now in the literature and to use methylation data to predict biological age. So as I said, that you could use different phenotypes to really create the biological age. But technically speaking, in the building of this epigenetic age, right? We need to really do a high dimensional regression analysis to create a prediction model. So the very popular method to do this to develop the prediction model is using the elastic net penalized regression. And this is the method used by Professor Haworth first. And then people follow this technology or this methodology to derive a prediction model and to make a prediction. Okay, so that's essentially a big contribution by a statistician, and then provide this toolbox to biologists who are able to use this GleamNet, our software, to develop prediction models, and then those prediction models are used to calculate the epigenetic age. So that's one side of story. The other side of story is how we're going to measure the physical activity, right? The availability of the various of the wearable devices provide the objective measurements of the physical activity. So now there are various of the devices available and used by people like Fitbit, Apple Watch, Actigraph, and so on and so forth. And for this particular data analysis we're doing, we use this the Actigraph, and that's the device with a sensor, a GT3XBT, that has, I think, 32 hertz. The functionality, basically this sensor measures 32 data points per second. Okay, so this device is relatively cheap, so that you can put this device on the study participants with very low cost. Essentially, that you need to really develop some like movement counts per epoch. Basically, you instead of looking at the raw data like 32 measurements per second, you define a unit of time, namely epoch. for example, 1 minute, 30 seconds or something like, then you basically count how many movements, right, within that time unit. So essentially, this sensor would track the movement by measuring acceleration. In principle, that this sensor will measure the, acceleration or movement in 3D sort of situation, right? So you can have X direction, Y direction, and Z direction. And then so essentially this if you look at raw data, you essentially get a tensor, right? Because this is 3D movement plus a variable of time. So it's four-dimensional data. But this created tremendous sort of difficult for practitioners to really handle this sort of tensor data. And what they did is that they do the dimension reduction. So they, create a summary sort of the data, like called vector magnitude, VM, which basically take Euclidean distance, right? So the square root of x squared, y squared, and z squared. So they basically convert a three-dimensional movement into a one-dimensional movement, one-dimensional sort of functional or time series data that you get out of this, right? So this is the one example of such, the sort of the VM, right, vector magnitude. So from 20 over 24 hours. Okay, so you can see that in the middle from midnight, like 0 time to 10 a.m. That those are the relative low activity time window, and then you have more activity in the evening, and in the after, more morning after 10 a.m. and in the afternoon. So here is the number of movements per minute. Okay, so in literature for the study of physical activity, there are many ways of dealing with those spikes, right? So there are a lot of wiggly, the time series looks very wiggly. There are a lot of spikes in this. So one of the approach people take in practice is trying to discretize this sort of time series of movements. And then, for example, they put people in the sort of a kinesiology lab and let people to have a different level, let people wear the device and do different type levels of activity, like slow walking, fast walking, running, and running fast. So that under different situations that they measure how they this the physical activity will look like, right? How the time series will look like, so that they can define this sort of cutoffs, right? So sanitary, light, moderate, and vigorous activity. So those cutoffs, right, indicated by the red lines, are actually derived from experiments in some labs where they recruit individuals to perform different levels of activity, so that the document those cutoffs, and then those cutoffs used by many applied scientists to really simplify the analysis. Instead of analyzing this sort of time series directly, then they basically just discretize it according to those cutoffs, and they can simplify the data in very, greatly, so that they can really manage the data analysis, right? But we know that this the wearable device data can be produced by different types of the wearable devices, and then you have Fitbit, you have Apple Watch, or you have iPhone, you have Actigraph, and so there are many different devices, and the different ways that the wearables can be worn, and then there are a lot of heterogeneities in the use and wearing of the devices. we generate a lot of batch effect and generate a lot of heterogeneity in the data during the data collection. So that having fixed the cutoffs suggested by the whatever the lab sort of experiment may not be directly applicable to a more broader and heterogeneous population, and even different type of use, different type of device. So there is a great interest in the study of physical activity in the smart health sort of literature. How do you sort of create a data driven or data adaptive cutoffs to replace this kind of static fixed cutoffs in this kind of data analysis? So that's one problem that we encountered through our collaborative projects and the University of Michigan. So how do you really derive, kind of data adaptive cutoffs to define the physical activity windows in a data-driven way? And then that's certainly a problem that we like to solve and to help our collaborators to, to answer their questions. So what we did here is to, introduce this sort of occupation time curves. So what we think here is really, if you want to come up with a data-driven cutoffs, you can think that the cutoff line may move from the bottom to the top of this this curve of the activity or the time series of activity movements. So what we're trying to do here is to create a summary curve we call occupational time curve. I will show you how to do this in 10 seconds. So essentially this occupation time curve is trying to compute the percent of the physical activity at the level C, okay, or above. Okay, let me just give you a example. So if I place the red line here at 0, that means I have 100% of the time over which that I have physical activity at 0 or above. Okay, that's really just 100%. Okay, so here I map my the percentage of time or my occupation time curve value at this given cutoff C equal to 0 to a percentage of time at physical activity level 0 or above. Okay, of course that I can move the line up, right? So then I move this red line up to a higher position. Now, for example, at this place that this line is the cutoff is placed at 1,000 movements per minute, then I'm calculating what's the percentage of time over this time window that the physical activity is at this level 1,000 or above. So I can calculate, that gives me 0.95. Basically, that means 95% of time this person over this time period would have physical activity at 1,000 or above. Okay, then you can keep moving this up, right? So then you can generate a another curve as a summary statistic in terms of the percentage of time of activity at a certain level or above. So finally, that you can reach the top, then you can generate a sequence of these points, right? In the right-hand side of this figure. Of course, that this movement is continuous. So at the end of the day, you will generate a continuous curve like this. Okay, so if you do that kind of exercise for every sort of this time series of the movements, right? So that you can end up with a summary curve as this percentage of time of a certain activity level or above. So this is more like survival curve, but it's not survival curve, it's occupation time curve. And then each individual would have a different shape of this curve. And then, you can interpret this as, different scenarios of the physical activity. So you can have less active situations, then you have more active situations. The curve looks different. So that can reflect the different situations of or different types of the activity. So what you see here is that you basically summarize very wiggly and the time search with a lot of spikes, which are hard to deal with in data analysis, into a quite smooth curve, right? Which gives you a I don't know if this filtering is good or bad. At least I think that would be as a principle that you like to create something more you know contains more signal than a lot of noise, because those spikes may have a lot of noise. You don't know how to interpret those noise, right? In this figure, when what you're trying to do is trying to smooth out some unnecessary noise and to create smooth version of it so that you can have more definitive patterns that represent different types of physical activity. Does this really make sense? Now, it's really say that whether or not those curves carry the enough variability across different subjects. So here is roughly about 500 subjects. Then if you do this this exercise for every individual for a certain given a period of time, that you do see that this occupation time curves exhibit a lot of variability, meaning that people have demonstrate different physical activity cases, which can be used to do the analysis you like to do. Okay, so this essentially create a functional predict a functional predictor of functional x variable or independent variable. Okay, so that this curve can be used as the a regressor in your regression model. Okay, so this really it's come with the sort of in very natural sort of choice to do the functional data analysis, because of this occupation time curve is your predictor, so that that you'd like to run this scalar and functional regression. So Y would be your biological age. So whichever the epigenetic age you like to use to study the relationship to answer your question. And X is the occupation time curve, like this is monotone decreasing function, which I just presented how this created. Of course, that this is very standard scale on functional regression, and the beta would be the parameter of interest, right? So if beta is 0, that means this physical activity has no impact on Y. If beta is not, then you look at how this functional the occupation time of physical activity might affect your occupational time. So this is very natural choice. And then I did not include confounding factors, but in real data analysis, of course, that you will put the confounding factors in the regression model to adjust the estimation of your association parameter beta. Okay, so that's a very standard regression analysis that you can use existing R package to do it, right? So there's no problem that you can run this. So that's what we did, right? So you put this occupation time curve X into this scalar on functional regression. Y is the methylation epigenetic age. Then you run this. So this blue curve is what you typically get, because this R package will do this spline expansion of this, functional predictor, and then you do this sort of regression analysis. Very, very standard using the, spline-based regression technique, right? So that you get a very smooth curve, and this. So when we present this estimate curve, this smooth curve, the estimate of beta, to our collaborators, then they feel that it's very hard to interpret this curve. One argument they talk to us is really that they do not see this sort of critical window, like window of this, because remember that in the very beginning, what they typically do in this kind of data analysis is to discretize the physical activity into different levels of different types of windows of physical activity, this moderate, vigorous, and so on and so forth. But when you have this continuous curve as the estimate of beta, they do not directly see this kind of critical window, which is it's hard to interpret, for them to really understand the association between the physical activity and biologic aging. So what they really want is this step function. Okay, so the beta could be a step function, right? Like this. So you have certain range of this x, and then a physical activity level, then what is the corresponding sort of effect size of this level of activity. If the activity drops, and then what is the corresponding, the effect size of the physical activity on y. So they want really this step function rather than a smooth sort of beta. If you want to create a smooth estimate of the, the functional parameter, then you can use existing R package. There's no problem. But our collaborator is more interested to estimate a step function. Okay, but then this create a little bit challenge for us, right? And so first of all, how many windows we have, how many change points, the analysis would, the data would suggest, right? Then how do you, do the this kind of estimation of beta as a step function, right? So this is a new problem, like we need to solve for our collaborator. So essentially, you think that this is a clustering problem. How do you cluster your the coefficients into different subgroups, right? Each subgroup has its own sort of effect size on your outcome y. So then there may be many, many solutions we can propose, but we did one solution that I'm going to present to you. So this is a solution that is based on the simultaneous operation clustering and estimation. So essentially, we look at the the solution, the mixed integer optimization, and I use this kind of the constraint optimization to solve this problem. Okay. So let me skip this. So the basic idea is like this. Okay, I have my occupation time curve that looks like this. So what you're trying to do here is you use a layman definition of integral. So you cut this curve into very, very tiny interval, okay? So that this integral can be written as a sum of the, of areas of the many, areas under the curve. And here, you can cut this to 300 intervals or 500 tiny intervals, depends on what you like to do. But anyway, so for each very small interval, that you can assume that this beta is constant. Of course, there's an approximation, and then this the integral from the left point to right point of the given interval will be the area under the curve. So essentially, you turn this integral cap into a sum of the beta coefficients and area under the curve over j successive intervals, right? So what next step, what you do here is that you are going to do a fuse lasso, right? You do a fusion. Because you don't believe there are like 500, the critical windows. You believe that there are only a few like actual physical windows that are really associated with your outcome, right? Not a arbitrary number of windows that you use to do this mathematical approximation. So what you're trying to do here is you first divide them to very little intervals, then you apply this fusion to fuse those the small windows into a bigger windows, right? So that you can create this kind of step function and end. Okay, that's basically this workflow that we adopt in this analysis. I mean, you can come up with different ways to do this, but that's what we did. Like so do the division and do fusion, right? So then, of course, that the one of the motivation of to do that is because we have the existing R package that allows us to run this fuse lasso. Okay, so you have a Y, then you have this linear regression, and you want to fuse of those beta J's into subgroups, right? So you just do this, the fuse lasso. Okay, so one challenge of using this existing R package is that this area under curve, namely AIJ, are highly correlated. The correlation can be as large as 0.98, 0.97. It's really, really highly correlated. So if you do directly use the diffuse lasso R package, like the graphic lasso, this R package, you will get a lot of bias in estimation due to this high dependence among those area on the curve. So for example, here is an illustration. So you can, enough of the situation, you overestimate some window size, or you may underestimate some window size because you do not have the right estimation of the change point position due to the bias in the estimation. So this creates a little bit, the unsatisfactory answers using the existing software. So we're thinking that how we're going to find a way that can really overcome this biased sort of estimation, and so that you can have relatively accurate way to find the location of the change point, so that you can obtain this size of the physical active window to draw a conclusion, right? So the team has work on this, discrete optimization solution. This MIT group has several, quite impactful papers to do this kind of sort of error zero penalty method. It's not exact error zero, but very close to that to solve the constraint optimization. So we had that kind of similar problem that they're trying to provide solution, so that we give it a try how that their method might work. Okay, so this is really the motivation why we switch to do this mixture integer optimization. Okay, so what we're trying to do here is like this. Okay. So here is the problem that we'd like to solve to to achieve some fusion, okay, for the beta. So then you do a augmentation, okay. The augmentation would allow you to uh, introduce this sort of group label parameter. So the eta is a new parameter. Okay, we introduce. We don't have eta parameter in the original regression model, but this eta is introduced to achieve the clustering or the grouping we like to do. So in this case, with two groups, for example, right? So that eta can be either 0, 1. So this is an indicator parameter to indicate which group that the beta will belongs to. Individual beta parameter will belong to, right? So this is a situation of two windows. So then, this is the way that we're going to, solve the a mixture parameter situation, because beta is continuous parameter and eta is binary indicator, binary parameters, right? So you have kind of mixed sort of the parameter situation. Some parameters here are continuous parameters. Some parameters are, categorical parameters, right? 0, 1. So you need to solve this object function with a little bit more challenging situation, okay, than you do, okay. So back to the problem we like to solve. So you start with a hypothetical number of critical windows, let's say k equal to 3 clusters, subgroups. So now you say that, of course, the 3 clusters would be determined by 2 cutoff points, like C1, C2 are 2 cutoff points. And then for each sort of sub-interval, for example, from 1 to C1, all the coefficients will equal to this zeta 1. And in the middle window, okay, then from C1 plus 1 to C2, all the beta coefficient will fuse to another parameter, zeta 2, so on and so forth. Well, of course, you don't know Z1 and Z2. That's the parameters that you're trying to figure out, right? So you don't know C1, C2. That's the, the cutoff points you're trying to estimate. Basically, the parameters that you introduce to solve your problem. Okay? And then the way to find the cutoff points is using this sort of the labeling parameters. So if the beta 1 belongs to the first group, then the corresponding binary variable will be assigned to value 1, otherwise it will be 0. So you introduce those subgroup labeling parameters. And then, of course, that you over-parameterized everything. So you introduce so many augmented parameters in this whole optimization problem. You are not going to get any you're going to run into the identifiability problem, right? So you need to put a constraint here, okay? So for example, the constraint will help you to reduce number of parameters so that you can solve the problem, okay? For example, if the eta 1 equal to 1, then this constraint force that the corresponding beta j will equal to a common parameter zeta 1, which is sort of the center of the first, the average or the representative value or treatment, the effect size of first critical window. Then if your beta j belongs to the second cluster or second window, physical activity window, namely eta 2j equal to 1, then this constraint will force corresponding beta to equal to the second, effect size of that critical window, so on and so forth. So this constraint will help you to reduce dimensionality. Of course, there's a sort of additional constraint about the relative position of C1, C2, because C1 has to be smaller than C2, because that is the cutoff, appears before the second cutoff, C2, and so forth. So you put adequate constraint here, then you can, put everything in the Groby, this commercial sort of the solver, to solve this constraint optimization. And then currently that Python has quite a nice package that can run this optimization. So in the paper, also studied some of this selection consistency property, and this is a finite bound of the how the under which conditions that this sort of the solution can be achieve the selection consistency. So you can define the oracle solution, which is the solution that you might get when you know the true number of group structures, namely number of the critical windows and also the the cutoffs, right? Of course, you don't know that. If you knew that, so what would be your solution? Okay, then you have your MIO solution, the mixed integer optimization solution obtained through this complex sort of constraints that we set up. Then you can prove that the property, the probability of your MI solution and the Oracle solution, being equal, will increase to 1 when sample size increase to infinity, and then this finite bound is established under this sort of the minimum change of mean square error condition, and then due to time limitation, you can find the details in the paper how to interpret this C minimum and so recently we are also working on this post-fusion inference, which is more challenging. So essentially, you really want to understand how to quantify the uncertainty of the effect size, like beta coefficients. And so we propose this sort of this ensemble inference is basically mimicking the idea, the thinking of Fisher's thinking of fiduciary inference. And yeah, the I can talk about a little bit more if we have time. But anyway, that's the ongoing work. We haven't finished this paper yet, but I think we have seen quite an encouraging numerical result, and we also did the theoretical sort of justification for this idea. Okay. So now I just want to show you some numerical result. I'll quickly go over this. So of course that you could have some easy sort of cases and difficult cases in this simulation. So the being easy or being difficult depends on really the gap between different windows. If in the clustering analysis, if you have two cluster that to be very separated, right? Then you can easily to identify their label their labels. If these two clusters are very close or even overlap, it's very hard to get the right labels. So now in this case, we in the simulation we put a very big gap from this regroup situation. So right, so this is the height of first group, this is the height of second group, and this is height of third group. So that here is the two cutoffs. Okay, so then you have different sample size, then you have so different the method to do this, and then we could use the MIO solution. We can use the fuselage solution. You can say that use the MI solution, the solution is the bias is very, very small. But if you use the existing R package to run this, the bias is quite large. Then you can see that here is the solution from the red bar here is the true window sort of the true window, and this inside is the one from obtained by the MIO where you can estimate the cutoffs. Okay, and then these blue ones are the one directly generated from the existing R package. So as I mentioned already, that this one is challenged because the high correlation. Okay? So you can do a little bit more challenged situation where your window separation is small, and then it's much harder to estimate the to do the separation, estimate the right cutoffs, right? So now you can see that the MIO still gives, this is true cutoff. The MIO still give very good sort of estimate of the cutoff in comparison that this, diffuse Lasso using the existing package gives a lot of bias in the estimation, which is not really something that we would recommend to use in practice. So finally, I just want to spend probably a couple minutes to talk about application. As I said that in the beginning, that this whole project was motivated to study association between biologic age and physical activity. Okay, so here in this example, we look at the skin blood age from about 500 children living in Mexico City, because Mexico City is in a high evolution, and there's a lot of radiation, so that people are interested in the skin age among the children. And then they do physical activity, so outdoor activity certainly give them more opportunity to have the exposure to the radiation when they're living a city of high evolution, right? So there are confounding factors adjusted. Okay, so now we did this analysis with different window size, and then different type, the different numbers of clusters were physical activity windows. Then we use BIC to choose number of windows, and then here's the estimates, and we use different number of the division of the small windows, 360, and so on and so forth. So there are several sort of choices what you're going to set up your problem, the optimization. But overall, the conclusion here is that if the younger skin age tends to be associated with the less activity, outdoor activity. So that's essentially what we found. And also we found the cutoffs in the analysis. And the overall message here is the less activity tends to have younger skin age. Okay. So I want to wrap up my talk here. So basically what we have done here is a new development of supervised homogeneity pursuit for highly correlated signals. Okay. There we carry out kind of simultaneous operation clustering estimation using mixture integer optimization formulation. We do not do this two-step analysis. First do clustering, then estimation. So basically we're doing this subgroup or clustering together with the estimation of the effect size. Okay, so that's really sort of one-step estimation for change point and parameter values to avoid the propagation of operational errors in data analysis. And so this whole analysis achieves the primary sort of the goal that the collaborators want us to have the method to develop some adaptive ways to derive cutoffs, right? So those cutoff points or values are estimated from the particular data we have, rather than using this given values in the literature. So this is adaptiveness that we as part of our solution will provide to our collaborator. And then the simulation result looks pretty promising. And so that this analysis is quite a new, and then of course they use some more advanced technology optimization to achieve this. And anyway, so that's what we did. Of course, that there's a limitation in this application because we used the Groovy as the optimization solver to handle this high sort of this mixture of integer optimization. So our experience suggested that if you have 500 beta values and or so, then the group B can work. If you have more than that, then group B cannot work very efficiently. So then certainly we need more powerful computational solver to solve this mixture optimization problem. And anyway, this is really a bottleneck of this computational aspect of the proposed approach. Thank you for your attention, and yeah, hopefully that we can have a little bit more discussions and Q&A. Yeah.